Alrighty, I think we'll get going here. So good morning, everyone. Welcome to our third session in our genetics testing echo series. So great to have you all joining us. We do have one more part in this series. It's got postponed. It was supposed to be on July 10th, but it's not going to happen until July 24th will be the date for that next one. And we're going to wrap it up with a discussion on whole genome sequencing. So I hope you'll join us for that. Introductions, I'm Troy Jorgensen. I'm the program manager here for Project Echo Nevada. And we have Dr. Susanna Sorrentino joining us from the UNLV School of Medicine, who's going to be our speaker today. But first, I'd like to go around and have you all introduce yourselves. So, Kim Hart, could you start us off?
Sorry. Yeah, hi, I'm Kim Hart. I'm with the Utah Newborn Screening Program.
Thanks, Kim. Good to have you today. Cindy Adams? Oh.
Sorry.
Go ahead,
I was trying to do it. Sorry. Cindy Adams from Rural Mobile Crisis. We deal primarily with suicidal and behaviorally dysregulated youth.
Thanks, Cindy. Jacqueline Loya?
Hi, Jacqueline Loya with the American Cancer Society out of Riverside, California.
Thanks, Jacqueline. Good to have you. Annette? Annette, are you there? Could you introduce yourself? We did have a little bit of feedback from you.
How about now?
Go ahead, yep.
Is it better? I'm Annette Lauren. I'm the Mountain States Regional Genetic Network Project Manager.
Great, thank you. Next is T. Olson.
Yes, I'm a physical therapist from Nevada Early Intervention Services.
Great, thanks for joining us today. Christy Weiss, do you have audio? I think you said you're having some trouble hearing us.
Can you hear me?
Yep, go ahead.
Oh, yep, I can hear you better. Thank you. I'm Christy Weiss. I'm from Mountain States Regional Genetics Network as well, and I am the Consumer Engagement Director and Social Media Coordinator for Mountain States Regional Genetics Network, which is an 8-state region in the Mountain States area. Thanks for having us, and thanks for letting us partner with you on this.
Yeah, thanks for all your help on this, Christy. M. Rindler? If you could unmute yourself with the microphone icon in the lower left corner. Go ahead.
Can you hear me now?
Yep.
This is Mary Rindler with the Utah Newborn Screening Program.
Thanks, Mary. Great to have you today. Carla Ortiz. Actually, Carla, it doesn't look like you have a microphone today, so if you just want to write in through the chat box and let us know who you are and where you're from, that would be great. Next is Nicole. Nicole, it looks like you have a microphone that's unmuted. Can we hear you? Yeah, go ahead.
Can you hear me?
Yep.
Okay, I'm Nicole Sweeney. I work at in an outpatient pediatric office in Reno.
Thanks, Nicole. Good to have you today. Dr. Robin Palmer, are you there? Do we have audio? No mic for Dr. Palmer. So next we'll go to Kat McPherson. Looks like you're joined by the phone, Kat. So it's *6 to unmute yourself. Go ahead. Oh, there you go. Go ahead, Kat. Yep.
Yes. Good morning, Kat McPherson. I'm with Amicus Therapeutics. Thank you for having me.
Thank you. Good to have you today. Jasmine T.
Yeah, good morning, everyone. Jasmine Torres from the Newborn Screening Program at the Nevada Public Health Lab.
Thanks, Jasmine. Good to have you today. And then Dr. Michael Koss.
Hi, radiation oncologist, Reno, Nevada.
Thanks, Dr. Koss. And then Robin Palmer, PhD certified genetic counselor in private practice, Northern Nevada Genetic Counseling. Thanks, Dr. Palmer. So I'll turn it over to Dr. Sorrentino for today's talk. Feel free throughout the talk to unmute yourselves if you have a question or comment, or write in using that chat box. I'll send something out in the chat right now so everybody can see where it is. It'll pop up like a little orange bubble for you. So feel free to write your questions or comments in through that as well. Alrighty.
Okay, great. I'm assuming everyone can hear me. Thank you all for joining us today and making time to join us for this presentation. Today, I'm going to talk a little bit about clinical genetics and its applicability to the clinic and to medicine, and then talk more specifically about genetic testing and why it's important and how you can use it and how you can even order it as a non-genetics practitioner. So first thing we're going to talk about is what is clinical or medical genetics exactly? I get this question asked a lot. I think there's confusion with some people as to what it is. Basically, a clinical geneticist is an MD physician who applies genomic discoveries or applies discoveries regarding the genome to clinical practice. And MDs, obviously like every other MD, goes to medical school, does a residency, mostly in a primary care setting, mostly pediatrics, but sometimes even internal medicine and obstetrics, and then does a fellowship in genetics. So we often also work with, bench research people or people who are not involved necessarily in medicine. And that's the research aspect of our job or of our role. So next slide. So what does the clinical geneticist do, or what types of patients do we see? Like primary care physicians or the primary care team, we really can come across almost anything. Everything sort of comes our way. It's not sort of system-based. So most consults are based on rare conditions, but we can also see patients with common presentations. And again, virtually any organ system can be involved. Although most of us are trained as pediatricians and most of us see the majority of our patients are pediatric, we are trained in seeing both adult patients and patients who have an individual or a family history of cancer, as well as preconceptual or prenatal patients. Next slide. So what are some of the reasons for a genetics consultation or referral? And this would apply to you as far as when you're in your clinical practice, what are some of the things you should be looking for that might flag, hey, this might be a genetics patient, or this may be a patient that requires genetic testing. And like I said previously, it runs the gamut, but I'm going to go through some of the highlights, some of the sort of more major reasons why someone may be want a consultation from genetics. So the first thing is unusual stature, whether it's very tall or very short, especially in comparison to the patient's genetic background or the patient's family's stature, so the stature of the parents. If you have 6 or more cafe au lait macules, and this is especially true, for people we suspect that may have neurofibromatosis. Something like a statin-induced myopathy or any other concern for how a patient may respond to drugs. And this sort of gets into the realm of personalized medicine, but certainly there is now genetic testing for certain conditions as far as how that person may respond to certain medications. Unexplained or elevated CPK. These patients usually first, obviously, the primary care physician will rule out a trauma, some infectious disorder or rheumatological cause, but once all those have been sort of ruled out, people start to think of inborn errors of metabolism, such as glycogen storage disease, and then that would be a referral to genetics or a flag for a genetic disorder. Next slide. So this next one is a very important one that we see a lot. This sort of falls under the general umbrella of neurological disorders. A lot of neurological disorders have genetic origins, as you might imagine. And one thing we see a lot, or we get called about a lot, is unexplained or recurring seizures, and especially those that are unresponsive to conventional management. And in addition, especially those seizures that are now coupled with other symptoms, such as unexplainable dystonia or muscular dystrophy, etc. Any excessive bleeding or clotting, especially recurrent deep vein thromboses or pulmonary emboli in younger people or young adults. And if the patient has one or more birth defects. And for this last one, I'm going to go into some more detail about birth defects, especially since we're mainly focusing on a pediatric population as far as what type of testing we could order for that. Next slide. So going back a little bit to the neurological conditions, besides the unexplained seizures, things such as unexplained myopathy, early onset dementia, or any other type of psychiatric disease for that reason. And then, of course, the movement disorders, you know, dystonia, choreoathetosis, etc. And then certainly we get a lot of these referrals, especially at UNLV. We have our own cardiogenetics clinic here. Anytime you have a patient with a cardiomyopathy or a conduction disorder, such as, you know, Brugada syndrome or early onset hyperlipidemia. And then these also, these types of clinics also, we also see patients with a suspected connective tissue disorder, which sort of ties into the cardiovascular aspect. So people with, you know, dilatation of the aorta, especially the thoracic aorta, or other dilatation or issues with multiple or major arteries, obviously anybody with poor wound healing or hyperextensibility, any tall stature, anything that could indicate a connective tissue disorder. Then people with vision issues, especially unusual vision issues, and you're going to be sort of hearing this over and over, unusual, different, sort of doesn't make sense. These are the underlying themes of genetic disorders. Because they're rare, they're difficult to diagnose, so you'll hear these themes over and over. So for familial hearing loss, you see a pattern of retinitis pigmentosa or early onset macular degeneration. And when I say early onset, you know, someone in their 40s or 50s. Early onset cataracts, especially in newborns or young babies, that's a flag for something genetic. Hearing loss is pretty, you know, pretty wide, pretty much runs the gamut. Most people with hearing loss could warrant genetic testing. Obviously, when we evaluate people for hearing loss, we're looking for syndromic versus non-syndromic, and that indicates or that guides what testing we order. Even for those that have non-syndromic hearing loss, there are several panels that you can order to determine, you know, whether it's autosomal dominant or recessive. And in some cases, by identifying the gene, you can correlate, or you can put together a genotype-phenotype correlation, where in some cases you can get a sense of how progressive the hearing loss may be. If you recognize a genetic disorder in a patient, so for example, you know, some of the more common ones like Down syndrome or Williams syndrome or 22q11, you know, if you see some of those, or for example, if you have a flag for a positive newborn screen and the patient has been diagnosed with a metabolic disorder or an inborn error of metabolism, that patient would benefit from a genetics consult or for seeing a geneticist. And what we have here at UNLV is we have a lot of patients coming either who suspect, the physician suspects that the patient has this diagnosis, or the patient has already been diagnosed. And many times what we do, there are well-established criteria, especially for things like Down syndrome or 22q11 or DeGeorge syndrome. There are very well-established criteria. And so therefore, as part of our evaluation, we talk about what specialists the patient might need, what, you know, diagnostic studies, what follow-up the patient may need. And a lot of times the patient will come for one visit where we go through the full evaluation, we go through what the care might be, and this of course all goes in my report. And then we also conduct genetic counseling, and we also conduct prenatal or preconceptual counseling for the parents, and then we arrange for genetic testing for the parents as well. And then a lot of times these children, except for probably the inborn errors of metabolism, they feel most comfortable going back to their pediatrician, sort of being managed in that way, but that's a possibility. And then one thing that often surprises people is mental illness or psychiatric disease. What we're finding more and more is that many individuals with schizophrenia, depression, bipolar disorder, other psychiatric diseases have an underlying genetic condition. Many of them are what we call microdeletion and duplication syndromes. And we're going to go into some detail in later slides as to what those are. I tend to diagnose a lot of these patients when I get referrals for fetal alcohol syndrome. A lot of patients who have been exposed to alcohol, amphetamines, or other illicit drugs during pregnancy have parents who obviously also are addicted or do have those concerns, and those parents in many cases also wind up having a microdeletion or duplication syndrome. So that's surprising to a lot of people, but actually winds up being the case.
Okay, so we do have a question that came in. Do you see adults with joint hypermobility?
Right now in my practice, because we're so swamped, I do not see adults in my practice. We're only limited to children.
Okay. Do you know anybody that does see those adults?
Yes. There is a doctor in Phoenix, Arizona. His name is Derek Nielsen, Nielson, I think. He's a clinical geneticist, MD, and he has a Ehlers-Danlos hypermobility clinic in Phoenix.
Great. Thank you so much.
You're welcome. And so if you're looking for conditions, if you see a condition that involves one organ system with one birth defect in one organ system or multiple organ systems, and when those things are unexplainable or unusual, you can't sort of seem to tie them together, or they don't fit together, or what geneticists affectionately call the $1 million workup. This patient has seen virtually almost every specialist, has had every diagnostic study, and people cannot figure out why, what this patient has, or sort of how to guide care or what to do. That's a flag that this patient probably needs genetic testing or needs a genetic consult. And certainly if you have a relative in the family history with sudden or unexplained death, particularly at a young age, so someone in their 20s or 30s that has a cardiac death, or a SIDS patient, many of the SIDS patients we now know can have underlying undiagnosed metabolic conditions, for example. And then if you do a family history as a primary care physician or on the primary care team, and you notice a pattern on the family history, so that you have multiple relatives with similar symptoms, especially first-degree relatives, but even relatives, in different generations. And then briefly, I know that there was certainly a cancer lecture that was done before, but the other flag would be for cancer syndromes. If you have an extensive family history of a single cancer, meaning in multiple generations you find the same cancer. So a good example of that would be breast cancer, isolated breast cancer. That's pretty common in the mother, the grandmother, and the great-grandmother, or the great-grandmother's aunt, the great-grandmother's sisters, for example. That's a flag for a possible genetic syndrome. Or if you have different cancer cluster types. So cancer cluster types include, for example, the more common BRCA1, which could be like breast, prostate, and colon, or a TP53 cluster. And if you see that occurring in 2 or more first-degree relatives, that may also be a cancer syndrome diagnosis. If there is an unusual cancer for the patient's age, so for example, if we go back to breast cancer, if you have a patient who's developed breast cancer at 25 years of age, that's early. So that may be an underlying genetic condition. And then any strange or unusual cancer presentation that can be indicative of an underlying syndrome. So a very rare or unusual cancer that may be also indicative of a genetic disorder. So we're going to shift gears now and talk a little bit about genetic testing. And within these next few slides, I'm going to describe the genetic test and also try to give you a sense of maybe how to read it or how to understand it, so that if you did want to order it for your patients and you had a result and maybe you were waiting for a consultation from a geneticist, you can sort of have a good sense of what to tell the patient, but also to give you a sense of when to order the test. That's also important. So the 4 types of tests we're going to go over are sequencing, chromosomes, chromosomal array, and then talk about metabolite testing for inborn errors of metabolism. There are other genetic tests that we can talk about, but I think these are the more useful and the more common ones, and probably the easier ones to order as well. So the first thing we're going to talk about is sequencing. So basically, in a most basic sense, sequencing is basically trying to figure out the order of the DNA. So if we hark back to our medical training, we remember that DNA is made up of nucleotides or bases, and these bases are given letters, A, C, G, and T. And that makes up an organism's DNA. There's over 3 billion of these, and so you can sort of keep that in mind when your patient asks why it takes so long to get a genetic test result. Depending on how many of these billion base pairs or nucleotides they need to sequence, that's why it takes so long. Next slide. And so this is a depiction, very sort of basic and rudimentary as to what happens when sequencing occurs. So without getting into too much detail, basically what happens is the DNA strand is unwound and 4 reaction mixtures are prepared so that each nucleotide is tagged with a color. And those nucleotides are separated according to color. And there are primers and other things that are used. Again, there's a lot of technical detail with PCR, et cetera, and amplification. But the basic point is that you separate these products on gel electrophoresis, and then you run them through a machine called a sequencer. And what the sequencer does is actually analyze the colors. And by analyzing the colors, it basically gives you the actual sequence or the order of the DNA, either of the entire genome or exome of the individual or of a particular area of the genome. And that's what sequencing panels are. So when someone says, I'm ordering a panel, what they're doing is they're not looking at all of the patient's DNA or all of the exome or genome. They're looking at certain areas or certain genes that have been pulled out and separated to be tested for based on the patient's symptoms. That's what a panel is. So when you hear about a panel or a sequencing panel, they're conducting this test, and what they're doing is looking at certain genes. Contrast that to something called whole exome sequencing, and what they're doing is they're looking at all the genes that are known to cause disease or known to cause genetic disease. Okay, so, or known to affect a protein or an enzyme. And then when we talk about whole genome sequencing, what we're talking about is sequencing all of the DNA, both the exome, both the exons and the introns, all of the DNA. Next slide.
So there was a question, how long does it take for a non-urgent referral to be seen by you, Dr. Sorrentino?
So for me, the rate limiting step is gathering medical records. So for example, I have appointments available in July. But it's difficult to get patients in because there's a lot of records, and staff, it takes a long time for the staff to gather the records. So what we tell patients is if they can gather the records themselves or if they have the records available, then it would just be no more than a couple of weeks. But other than that, it may be a month or more by the time we're able to collect all the records.
Thank you, and thanks for the questions, everyone. Please feel free to keep sending those in through the chat box.
And so now we're going to shift to chromosome analysis. And chromosome analysis is very familiar to many people. It's basically a laboratory procedure where you isolate the white blood cells from someone's blood. You break open the nucleus, and inside the nucleus is the patient's DNA. And what happens with that DNA is that it's spread out and there's a picture taken of it. And there are people who analyze how the DNA looks, the size and the shape of the DNA in order to determine whether there are any pieces that are missing or extra or translocated, meaning a piece is missing from one place and then stuck on another chromosome. So if we go to the next slide, it's true when they say a picture is worth 1,000 words. The picture sort of illustrates things. So when you order a chromosome analysis, most labs will not only give you the report, but give you a picture similar to this and show you the actual chromosomes. So there are some things I want to go over about chromosomes. Number one, the most important thing is that there should be 2 copies of every chromosome number, one from your mother and one from your father. And when I scan a chromosome analysis, that's the first thing I look for. Are there 2 copies of every chromosome. The chromosomes are numbered 1 to 22, and then X and Y if it's a male patient, or X and X if it's a female patient. And one thing you'll note about the chromosomes is that they're all different sizes. Some are bigger, some are smaller. The shorter arm of the chromosomes in a chromosome analysis report are reported as P. So P means petite. If it's the long arm, then it's reported as Q, or the longer arm. And then the light and dark bands have all subnumbers next to it, and that's important. And when we talk about chromosomal array, we'll go into detail about what that means. And so what you'll notice from this slide is that on chromosome 21, there's 3 copies instead of 2. So this is the classic presentation you'll see in someone that has trisomy 21 or Down syndrome. And I also want to point out that in the lower right-hand corner, the sex chromosomes, the X and the Y, are also very common areas of the DNA, or very common areas on chromosome analysis where you will see abnormalities. You may see multiple X's, you may see multiple Y's, or missing X or missing Y. But basically, this is someone in the laboratory who has been trained over many years to look at these and say, is there a piece missing? Is there an extra piece? Or is there a piece that is tacked on somewhere else where it shouldn't be?
Okay, some more questions about referral. So when referring a youth from social services that is FAS and has possible markers for Prader-Willi syndrome, would you just need a referral from a primary care doctor?
Yes, you need a referral from a primary care doctor, but if the child is in custody of DFS, or if their parents do not have custody, then the social worker would have to arrange, the social worker or the DFS worker would have to arrange for the consult and be present at the visit.
Okay, so a patient can't self-refer?
The patient can self-refer in the sense where the patient can say to their pediatrician or some other doctor, I want to see genetics, but we need an actual referral from their physician or someone in the medical field. And that's for insurance purposes, basically.
Got it. Okay, I think that answers those questions.
Okay, so next we're going to move to chromosomal array, or chromosome array. And how is that different from chromosome analysis? Instead of breaking open the nucleus and actually looking at the DNA, we're going a little deeper and looking for pieces of DNA that are extra or missing that you cannot see under a microscope. So they're teeny tiny. So they're called microdeletion or duplications. And so this is a sort of very short explanation about what array is, but I'm actually going to go to the next slide because I have a picture and I think that explains it, that is easier to understand. So what happens is that we take the patient's sample, the patient's DNA, and we label it with a dye. And in this case, the dye is red. And then we take the reference sample, which is basically what we know to be the quote-unquote normal human DNA sequence, or the normal human, in this case, the normal human DNA dosage, meaning, the normal number. And we mix those two together and we hybridize them on a slide. And then on the slide is run through what's called a scanner, again, analyzing color. And based on the variations in color, the computer can analyze whether the patient has a teeny tiny piece of DNA anywhere on the genome that's extra or deleted. And that's basically how we see that, or how we detect that without actually looking under the microscope, because some deletions and duplications are too small to see under the microscope, and this is how we detect it in that way. Next slide. Okay, so this is an example of a chromosome analysis report, a chromosomal microarray report. And so if you wanted to order the tests, I wanted to distinguish first the difference between a chromosome analysis and a chromosomal array. So when you order a chromosome analysis, it should say chromosomes, or chromosome analysis, or it can say karyotype. And when you circle that or you order that, you're ordering the first test that we talked about. If you see chromosomal microarray, or array or SNP array or pediatric array. Those are all chromosomal microarrays, which we're talking about here. Microarrays, like chromosomes, can be ordered both in pediatric and in adult patients, but also in prenatal patients. It's important to distinguish between the two when you order the testing, because when it's prenatal, obviously you're either sending placental cells if you're doing chorionic villus sampling, or you're sending amniocytes if you're doing amniocentesis. Once the child is born, obviously you're sending blood. So I picked this genetic test to go over because unfortunately for this patient, he illustrated basically all of the things that you could see on a microarray report. So one thing I want to call attention to is under CMA summary, the last line says genome build. So what the genome build is that at the National Institutes of Health, we have builds, or what we call quote unquote normal sequences or normal arrangements of what a normal human would have. And they're given these numbers. And these builds, these genome builds, are updated periodically, and the laboratory uses them to compare the patient's DNA to the quote unquote normal build or the normal DNA. The resolution, as you see there on the right, is basically how much can we sort of focus? How small can we get? How small can we see these either microdeletions or duplications? And in this report, it's 1.15 kilobases. So it can vary from person to person. So if we sort of pan out again, Troy, what we see here under CMA nomenclature is that ARR means an array. So they've done an array. The HG19 tells us the build, which we discussed above. And then it's telling us that on chromosome 22, on the long arm, we discussed before, short arm is P, long arm is Q. If you remember the light and dark bands that we saw in the chromosome analysis, and they all have numbers, each of them have sub-numbers. So at this sub-number, 11.21, at the exact nucleic acid position that's indicated here, there is only one copy. So it says ×1. So we talked before that you always want to see 2 copies of chromosomes, or 2 copies of pieces of chromosomes, one from mom and one from dad. So if it was normal, it would say ×2. Since it says ×1, that means we have a missing piece of DNA, very small, within those parameters. If it said ×3 or ×4, then we know we would have a microduplication or an extra piece of DNA or multiple extra pieces of DNA. So that's how you read the report. And this long, written indication as to where the deletion is otherwise known as DeGeorge syndrome. So someone with a deletion in this region is the 22q1121 region, has DeGeorge syndrome. And what I want to point out to underneath that is that there are many other areas listed here. For example, the first one says 5q31, et cetera, et cetera. And what you notice is that it says ×2. So you'll say to me, okay, Dr. Sorrentino, you said ×2 is normal. He has 2 copies. So why is this on the report? It's on the report for another reason. Next to it, it says HMZ. HMZ stands for homozygous or homozygosity. What that means is that the genetic contribution in this region from the mother and the father is exactly the same. So depending on how many regions are listed here, there can be a degree or a level of consanguinity. Obviously, if the regions are bigger, I unfortunately have had cases where I've had children come in and they have regions as big as 32% of the region being homozygous, and that was a father-daughter pairing. You can have uncle and niece, you can have brother and sister, you can have second cousins, third cousins, first cousins, it runs the gamut. So a lot of people ask me, well, why is this reported? Why do we need to know this? Well, we need to know this on a couple of levels. Number one, Obviously, if there's, and this I've unfortunately had to deal with, if there's an issue with incest or sexual abuse or rape, it may come out in this way, especially between family members. Secondly, the more regions of homozygosity that you have, the more likely that you will have an autosomal recessive disease. And so what I do in these cases is when I see these large regions, I analyze them to see what recessive diseases there may be in there. And depending on the level of consanguinity, you're more likely to have a recessive disease. And in order to get to what recessive diseases may be in here, what you have to do is do sequencing, which we talked about above. You have to sequence all these regions and see if there's any, if there are any recessive diseases. So this test does not pick up, so these areas, excuse me, are not microdeletions or duplications, but they're a screen or they're a flag that there might be a recessive disease.
Okay, so we do have another few questions that came in. Do you need any blood work prior to referral?
It depends. I don't. If you're comfortable doing it, if you're comfortable doing the microarray or the chromosome analysis before you refer, then go ahead and do so, but I don't. It's not required for me.
Okay. For a child with developmental delay or symptoms of autism, when would a genetic referral be appropriate?
So I'm actually going to get to that in a few minutes.
Great, okay. And then, let me find the other question here. Could you comment on the use of chromosome analysis in these days of microarray? When is chromosome analysis requested instead of a microarray?
So that's a really good question. So nowadays, microarray has largely, not totally, but largely replaced chromosome analysis. Most times, the only time I really order chromosome analysis would be for example, a blatantly obvious person with Down syndrome, who I'm pretty sure I'm going to get a trisomy 21, or if I suspect a translocation. So if I suspect that a piece of DNA has been tacked on to another piece of DNA, then the only way you can see the translocation is through a chromosome analysis. And the translocation may flag or pop up in the microarray, and then I go back to chromosome analysis to confirm it.
Okay. And then do you discuss the report that reveals consanguinity with the patient, or do you report it to the authorities?
So you report it, from my understanding, you have to report it to the authorities if the mother of the patient is a minor, or if when the incident occurred, in other words, when the conception occurred, the patient was a minor.
Okay.
And so therefore you'd have to report that.
Got it. And then Jacqueline, did I see you take yourself off mute? Do you have a question?
No, I'm sorry. I got disconnected, so I had to log back in.
Okay, no problem.
Okay, so now we're going to talk about what a lot of patients in the pediatric, a lot of providers in the pediatric field, so pediatricians and others, want to get a sense of. So if I wanted to order genetic testing, how would I know what to order, or where can I go to find out? So the American College of Medical Genetics and Genomics, who is the professional board that certifies me, has something called ACT sheets. If you go on their website, you'll see that. And the ACT sheets give information for certain types of presentations as to what testing can be ordered. And specifically, I'm going to go into some detail in the next few slides about developmental delay, autism, learning disabilities, intellectual disabilities, etc. The other thing that website puts together very nicely are ACT sheets for inborn errors of metabolism. So as a general pediatrician, if you get a positive newborn screen for a particular diagnosis, you can go to the American College of Medical Genetics and Genomics, and they have actual ACT sheets according to disease or according to the result on the newborn screen. So for example, say the newborn screen says elevated C3. You can go to the ACT sheets and they'll give you elevated C3, and then they'll give you all the possible diseases and what the workup might be and what the steps are, which of course in most cases includes contacting a geneticist for further workup. And then, thank you. sorry, go ahead. Yeah. So then, so what are some of the more specific things you look for when you're examining a patient, or especially in the pediatric population? What are some of the things you look for that might flag you, yeah, I need to order some genetic testing? If you have a patient or a child that has one or more organ systems involved, so for example, say the child has a heart defect or a heart defect and a renal defect, that warrants an array, a microarray. Or say, for example, you see dysmorphic features in the child. If you start to see dysmorphic features in the child, and if the child has one or more organ systems involved, that's definitely, you should definitely consider a microarray. Most of the time it would be a microarray. I say plus or minus chromosomes because obviously for things like, you know, if you suspect Turner syndrome, you could do chromosome analysis. If you suspect Down syndrome, you could do chromosome analysis. But the microarray will also pick up Down syndrome, will also pick up Turner syndrome. So, you know, if you weren't sure, it would be reasonable, you know, just to get a microarray to start. And then I wanted to talk specifically about developmental delay, intellectual disabilities, autism, and I'm sorry I didn't put that here on the title. Autism spectrum disorders also fall under this list. The American College of Medical Genetics and Genomics has specific guidelines for patients with these symptoms. And what they recommend is for all patients who have these symptoms, a microarray, Fragile X testing, and inborn errors of metabolism if needed. Meaning, if you're not sure about the newborn screen, for example, I may have older patients that are 14 or 15 years old that had newborn screening in another state, or no matter what state they had it in, you know, the newborn screening 14 or 15 years ago was not as updated naturally as it is today. And so if I'm concerned that there's an inborn error of metabolism that may not have been picked up years ago, I may do a workup. So on the next slide, I'm going to go over what that workup might be. When you do a workup for an inborn error of metabolism, what you're not initially doing genetic testing. You're doing metabolite testing. And what you're doing is you're looking to see how the patient processes the food that they eat, meaning proteins, fats, and sugars. So for example, plasma amino acids analyzes how someone breaks down proteins. And so it gives you a listing of several amino acids. When you're checking urine organic acids, you're looking for abnormal metabolites that are spilled into the urine when someone doesn't process either proteins or fats in the appropriate manner. An acyl carnitine profile analyzes free fatty acids, so again, fats and how people process fats. Total and free carnitine is related to fatty acid oxidation, but also to the mitochondria and how it functions. When you screen for ammonia and elevated ammonia, you're screening for disorders of the urea cycle. And specifically those urea cycle disorders that occur at the beginning, like OTC. On a Chem 14, you're looking for acidosis, so a CO2 of less than 17 or so. You're looking for low glucose or hypoglycemia, and you're looking for elevated liver function, and you're looking for an elevated anion gap, which would be greater than 14. Lactic acid and pyruvate is also important. And uric acid is a screening tool for glycogen storage disorders. As I said before, because these things are metabolites, they are an excellent screening tool, meaning they have a high pickup rate, but they're not necessarily diagnostic. So as we've all come to understand, especially those of us who take care of patients, both in the clinic, but maybe in the neonatal ICU or the pediatric ICU, these metabolites can be affected by diet, how long the patient has been fasting, whether the patient is on TPN, and whether the patient has an immature liver. So for example, I often get referrals for preemie children or preemie babies where they keep getting positive newborn screens for tyrosinemia because the metabolites that they have in their serum flag for tyrosinemia. But what it actually is because they were born at 28 weeks, they have an immature liver and they're producing these abnormal metabolites, but they don't in fact have tyrosinemia. So it's important to sort of realize that these certainly are very good screening tools, but they're not in any way diagnostic. So if the metabolite screen is positive, so if you're seeing a pattern or seeing something, the way to confirm the test is to either test for the actual enzyme and or do the genetic test. If we can hark back to med school or we can hark back to our training, we know that DNA makes RNA and makes protein. And an enzyme is a protein. So if you have a DNA mutation, you can have a defective protein or a defective enzyme. And that's what's involved with these metabolic disorders. Now, for some metabolic disorders, the genetic etiology is not clear or is not well described, or the pickup rate for genetic testing is not high. In these cases, a diagnosis can be made by studying the enzyme function. You want to look to see, does the enzyme actually work? You know, what percentage is it functioning well? And that can give you a sense of, you know, what the disease is. So for newborn screening for Nevada specifically, if you go on the website, it says there are 31 core and 29 secondary conditions that were added you know, as per the recommendation of the College of Medical Genetics and Genomics and the March of Dimes. You know, a lot of those are metabolic disorders, but of course there are other disorders. You know, we all know about sickle cell and cystic fibrosis, etc. There's new testing that's going to be coming along for the following conditions that were sort of in the works. And these are for X-linked adrenoleukodystrophy, Pompe disease, MPS I, and spinal muscular atrophy. And these are being added because most recently there have been either enzyme therapies or other therapies, including gene therapy. So what we expect throughout the years is for more and more diseases to be added, as there has been a huge boom, I would say, over the past 10 years or so of developing therapies, whether they be enzyme therapies or gene therapies for many rare disorders.
So there's a question here. Is it possible for a child to have a negative newborn screen and still tray positive later with metabolic testing, such as mitochondrial disorders?
Yes, it's very possible. So I'm going to go over in the next few slides mitochondrial disorders, and it's actually right here, and I'll talk about how you can have a negative result on newborn screen. So I wanted to comment briefly on mitochondrial disorders and the different types and how one may pop up later as opposed to another. So there are two types of mitochondrial disorders. There are mitochondrial disorders where you have mutations in the nuclear DNA, so the DNA of all your cells. And then you can have a mitochondrial disorder where you have a mutation in the DNA of the mitochondria. So the mitochondria have their own DNA, and you inherit that DNA, that mitochondrial DNA, from your mother. Now, I say that with sort of grain of salt because there were some recent studies that came out that where children actually inherited it from their father. But what we know now at this point is that the majority of us inherit our mitochondrial DNA from our mother. So what's the difference between these two? If you have a nuclear mutation causing a mitochondrial disorder, then it tends to be autosomal recessive. And when it's autosomal recessive, it shows up in infancy. So you start to see increased lactic acid. You may see elevated alanine and other things on plasma amino acids. Your acylcarnitine profile may be abnormal. And so those are the things that you may or may not pick up on newborn screen. However, mutations in mitochondrial DNA, symptoms don't usually show up until later. So most of the mitochondrial disorders caused by mitochondrial DNA mutations usually show up later in childhood and sometimes in adulthood. And those have a maternal inheritance. So you'll see that all the children born to a mother with a mitochondrial disorder will also have the condition. And so with those, you also see a lactic acidosis onset, but you see a lot of unusual symptoms. So symptoms where, for example, they may have an eye disorder, they may have a cardiomyopathy, they may have pancreatitis. So in different organ systems, you're seeing strange and unusual presentations, and they don't seem to fit together. You may be looking at a mitochondrial disorder, especially if you see persistent lactic acidosis, both on comprehensive metabolic panel and also just by measuring the lactic acid. And so just to add a little humor to today's presentation, this is a doctor, this is an internist with his patient, and he's telling him, eat less, exercise more, and invent a time machine so you can go back and choose parents with better genetics. And then finally, I want to just remind everyone that despite everything, everyone is 50% their mother and 50% their father genetically anyway. And then for the last slide, there are some resources that I think are excellent and geared towards physicians for people that may not have had a lot of either experience or a lot of education regarding genetics. Both these resources are excellent in answering any questions you may have about genetics and genetic testing and referring, and also for the source on the left regarding personalized medicine, which especially in the adult population is an up and coming idea or concept. And that's it. So are there any other questions that I could answer?
Yeah, it looks like there's been a few that came in. So the other part of my question, telling a patient or parents that report revealed consanguinity, am I thinking correctly that percent of homozygosity will reveal you how closely related parents are, such as first degree relatives, but will not differentiate who father-child versus mother-child or brother-sister?
So if it reveals a first degree consanguinity, then it would be either a parent-child or a brother-sister. If it reveals, if it's a little less consanguineous or the percentage is a little lower, then it's probably an uncle, niece, or aunt, nephew, okay? And then after that, you would be first cousins, then second cousins, then third cousins. So yes, depending on the area of the DNA, it can tell, how much, the percentage, it can give you the relationship, but it can't obviously tell you the exact individuals, but it can narrow it down as far as their relationship.
Are all mitochondrial disorders inherited? Is it possible to have an acquired mitochondrial disorder from exposure to a drug, a chemical, or a toxin, such as a statin or Depakote?
Right. So technically, you can acquire, not just mitochondria, but you can acquire a genetic disorder. And we're not 100% clear on how that happens. That's the whole area that we call epigenetics. I can tell you the majority of the mitochondrial disorders that I've seen have not been what we call de novo, meaning have not popped up only in the child. They have been seen, we either identify that the parents are carriers and that it's recessive, or that it's been passed down from the mother. But technically, could it happen? Yes, it could happen. It's just not seen very often.
All right. Any other questions out there? Feel free to unmute yourselves or write in through that chat box. And while you think about that, I will share the screen and our next session. So it's a few weeks away, July 24th, but we're going to have our partners from the Nevada State Public Health Lab joining us to talk about whole genome sequencing. So that's our last one in this series. So we'll hope you join us for that one. I send you all reminders as well, so just keep an eye out for those emails coming from me. Just a thank you here from Christy Weiss. Love the presentation, well done. Thank you, Dr. Sorrentino, for your time. We hope to link back to the recording on our website. Yeah, we'll be working on getting these recordings made available, so folks, those of you who joined us today can watch it back, or anyone who might have missed it can watch it as well. So I think that's it for today. Thank you so much, everyone, for joining us. Thank you so much, Dr. Sorrentino. This was fantastic. So great to have you.
Great. Thank you. Thank you for having me.
All right. Have a great rest of your day, everyone.
You too.